And we'll speak about desynchronization in the development of GABA pathway in mice that expressing low activity of 1-carbon metabolism due to genetic, in this case, knockout, but representing actually a population of children where the MTHFR gene is polymorphic in the site that actually replace one amino acid in the catalytic subunits of the enzyme and then reducing the activity of the enzyme. So in human, it was shown that both deficiency or this polymorphic allele in the child in homozygotic way or in the mother increase the risk for autism. And also there are additional evidences that if the pathway in which this enzyme is active, the pathway that incorporates folic acid into the 1-carbon metabolic pathway, have low activity, either from genetic or other causes, and for example, problems in the gastrointestinal system or selectivity in eating or other reasons, antibodies against the folate receptor, for example. So low activity of this pathway is really predisposing the brain to the developmental consequences that lead to autism. So in previous study, we actually assessed the SD-like behavior in these mice, either if they are heterozygote to the MTHFR or the mother, or both, them and the mother, and we will show the scheme. And we show there that there is a correlation between, correlation between the behavior of the mice and the expression of proteins in the GABAergic system. And this scheme actually show the non-syndromatic behavior. They have the GABAergic synapse with sufficient amount of GAD, the enzyme that actually decarboxylate glutamine to GABA and enough vesicular synaptic vesicles with sufficient VGAT, whatever have the normal mouse, and all the postsynaptic elements, receptors, and so on. And in the syndromatic mice, we saw a reduction in the GAD and in vesicular GABA transporters, and changes, of course, also reduction in the NKCC1, a transporter that actually is critical part in the regulation of chloride concentrations in the postsynaptic cell, leading to changes in the reversal potential of the inhibitory synapse. So this was the previous work, and we based on this and other evidences that also show alteration in the GABAergic system in the adult mouse. And this is this part of the scheme. So our study framework is that these changes that happened early in life, they will produce, and some of it we showed, changes in the early behavior in the offspring, leading to SD-like behavior in the adult, and changes in the GABAergic system at early stage may be the reason for these late changes that we showed in the adult. So today we will focus part of the talk in the early age, changes in the GABAergic system that we are curious to see if they appear already early in life, and the changes in the adult GABAergic system that may hint to origin in the early stage. And what we are really looking is that to find at the early age what predictive changes we can detect behaviorally, and Itai can show you part of this work in his poster. If the changes in the early stage are already there in the GABAergic system or in other system, and that's Danielle will show in her poster, and I will show part of it here. And we hope in these mice that we can characterize what's happening in the brain and the risky or the risk in the behavior at early stage to be able to find later on molecular markers that we can detect in their blood, which will lead, hopefully, in the future to find some biomarkers for those that need intervention. So the objective of the study that I will show today is to, or part of it I will show today, is to find what is changing in the brain at early stages and where it's and when it's happening. So the three W's, and what is the impact of the parental genotype. We showed in previous work a very strong fingerprint of the maternal genotype, but due to suspicion of Noah, that she's the student that led this project before, she said, I still need to see how deficient offspring develop in normal uterus, because maybe the effect of the maternal genotype is reflected by the in-uterus environment, and if so, then we will see like that the development at the embryonic stage is normal. So we'll try to answer part of these questions here. So until now, we used to work with a strain of mice that express that the knockout was introduced to it is the bulb C mouse that have very low social activity in the baseline. And for the purpose of this study, we mix this strain with a strain that have a different background, the C57 background, like Evan showed in some of his models, which is much more social mouse. And this strain also, the colony that we have, express the red protein, TD tomato, in all the inhibitory cells in his brain. So it's also easier for us to analyze the inhibitory system. So in the study design here, we had 5 groups. Always the one to your left is the mother, the middle is the father, and the offspring in gray, which is the F1 of the same cocktail, but always the black mouse is carrying the wild type gene, and we don't have the opposite mix for the wild type group, which is the control of all of it. So some of the work that I will show today are our classical groups 1, 2, and 3 here, the upper layer, and all the experiments were performed on the 5 groups, but we didn't finish to analyze in some of them groups 4 and 5. So either 3 or 5 groups of this mix. So we'll see work that showing the autistic-like behavior in the adult mouse, 3 months old, and a few days later, these mice were anesthetized, decapitated, and we looked at their brains for the population of the inhibitory cells containing GAD, the parvalbumin and somatostatin populations of cells that are critical in some of the important processing done in the cerebral cortex. And in the early age, we looked at the same groups, looking for the early origin. And at this stage that we were like just looking for first signs, we began with the age of postnatal day 0, the day of birth, when whatever the mice had have a strong impact of the embryonic development, and 7 days old. That's it. So moving to the data. So in this study, I choose to look at direct interaction, not the 3-chamber that we added in all our previous studies. 2 mice were separated for at least 4 days to individual cages, and then they were transferred to neutral cage. None of them was familiar with this cage, and then we just videotaped and recorded ultrasonic vocalization. Let's stop them. They going to kill each other. No, they didn't. But Noah actually analyzed different types of interactions, sniffing, nose to nose, nose to anal genitals, crawling under, crawling over, biting, whisker trimming, and different type of interactions, positive and negative. I will show here for the short time, due to the short time, only the total number of interactions initiated by each of the the mice in the video. We felt that it's much more reflecting the natural behavior of the mouse than the tree chamber that is very, very good for many purposes. But here we just wanted to have a behavioral correlation to the other findings. So we saw that in the black line, the control group performing the largest number of interactions. And in green and red, solid lines are those that their mother was heterozygote to the gene, and in the dashed lines we see the impact of the father. So not only the mother, it's also the father. But I was really very, very skeptical when I saw these results, and I felt uncomfortable with this because the in utero ability, or the fact that previously we were able to correct the behavior of the majority of the mice by nutritional intervention during pregnancy somehow gave me the good feeling that it's something that we can correct during the time that the embryo is in the mother. But these are the results, and then we moved on to look at other features that we saw previously due to the maternal genotype, inhibitory cells in the brain, so all the red cells are GAD positive, and we see here examples of the two populations, the somatostatin and parvalbumin, marked by circles, work by Gilad and Noah. And when we analyze the number of inhibitory cells in the different layers of the cortex, from one superficial to the deep layers, layer 6, we saw that, first of all, we have less inhibitory cells in all layers of the cortex. Looking at the somatostatin expressing cells, we saw a decrease in the, which group? Wild type offspring that grew up in a mother, or is a son of a mother with deficiency in the MTHFR. And this is something that we saw also previously in some of the behaviors that somehow, and also in metabolites of the blood of these mice, that somehow the heterozygote, growing in heterozygote mother is more resistant than the wild type. And we are still puzzled about it. It's not in all the features. This is also the total number of cells, but less inhibitory cells in the wild type offspring. Looking at the parvalbumin population in the 5 groups of the experiment, so we see again major effect in the same group, but to the opposite direction, more parvalbumin cells in the layer 3 in this group, and some changes also in the deep and superficial layers, which are parental dependent, like behaving different in the dashed lines, father carrying or lacking the gene than the mother. Origin. So since there were reports previously in some models of autism in mice that there was a difference in the ratio between these cells, and since we saw the difference exactly in the same layer 3 of the cortex, we calculated the ratio between to see if the change that we see in these two populations is somehow related, and indeed we saw that we can, as evidented here, see an increase in the ratio between parvalbumin to somatostatin. And when looking at the different layers of the cortex, we can see that the two groups of offspring to heterozygote mother behave a little bit different in the layers that are actually affected. So this hints to very early origin of the deficiency, because the changes or the decision making of which is which, both type of neurons are generated in the MGE, medial ganglionic eminence, in very, very close areas. At the early development of this area, cells prefer to be the yellow ones, and in the later age of this, like 2 days later, if the cells are proliferated, they will favoring be the blue cells that we see here. So this is hinting to very early age. And in addition to something in the laminar distribution in processes related to migration. So we decided to go back to the early stage, and we collected the brains for these immunohistochemistry studies, which we are still processing them. But in the meantime, Daniel was fast enough to look at the proteins that we saw changing in the adult brain and to see what's happened to them at the day of birth. And surprisingly, what we see here is that the proteins that were changed in the adult brain, the VGAT and the GAD, are not changing at the early stage. And these are proteins that are present in the inhibitory neurons. The other proteins that we tested are present in all the inhibitory synapses because they are postsynaptic. And here we saw in all proteins, gefirin, the protein that actually hold the inhibitory receptors on site, KCC2, which is critical for the regulation of chloride, and if it's there at this age, it means that the synapses are already inhibitory. And NKCC1, which is expected to be there by the natural regulation, but we see, again, higher levels of this protein. So the ratio between NKCC1 and KCC2 is somehow suggesting that maybe NKCC2 try to compensate for the presence of KCC2, but KCC2 is not expected to be there until postnatal day 7, at least. All right there. And we see very, very strong expression, and it's something that Danielle repeat, and she'll repeat in more animals because it's really a very strong result. So if that's the case, it means that the inhibitory potentials at this age are hyperpolarizing, and in addition, there are evidences that the expression of KCC2 is one of the signals that attenuate and stop the migration of inhibitory cells within the cortex. So there is a link between these two signalings, and this is another evidence for facilitated development of these cells. So they mature maybe earlier than they are expected to, and if so, my prediction is that for valbumin that's supposed to be expressed only around postnatal day 9, we may find it already in these animals at earlier stage. So the changes in the inhibitory system are not completely synchronized because we see changes in the postsynapse and not in the presynapse. We see evidences for changes in decision-making or the fate of the proliferating cells if they are going to be somatostatin or parvalbumin, which is critical for the circuit processing. And we see evidences for some preconception, maybe germline changes due to the MTHFR deficiency, and it was tested in the fertility, at least, of these mice was tested, and there are some evidences that indeed something in the sperms is already changed due to the MTHFR deficiency, but there is still, we need to show that connection along the chain. So what we are doing right now, Itai is trying to figure out if ultrasonic vocalization may be one cue for, or predictive cue for later changes. Of course, we need to see how early all these things happened and what are really the changes. We try to, in the puzzling of excitation inhibition, we try to see if we can see some subthreshold evidences by EEG recording from the adult mice. So this is the upper part. And that's more all that I want to tell you today. To your left, the people that contribute most to what I showed today and what we are doing now, and to the right are past people that work on the model and collaborators and funding. Thank you very much.